Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video sessions from Biology. In a series of video, we are going to talk about the antiviral drugs in a detail. Whenever I was preparing that topic, antiviral drugs, whenever I preparing that topic, then I search some videos on it. So the video which are prepared on antiviral drugs are very short videos, and these videos are not important for whenever you are doing a PhD, master or bachelor. Whenever you are the student of PhD, master or bachelor level, you need to understand the mechanism of action of the antiviral drugs. You need to understand the resistance which are activated against these drugs and broad spectrum of these antiviral drugs and some other properties you need to understand. Fine. Now in this video lecture, I will talk the basic overview of the antiviral drugs or the big picture of the antiviral drugs. Once you understand the big picture of the antiviral drugs and once you get the idea about the antiviral drugs, then the rest of the videos which are regarded to the antiviral drugs will be very easily for you. But before going to explain this antiviral drugs, you need to understand the virus structure. You need to understand the shape, the size, structure, everything you need to understand. That you also need to understand the important component which are present inside the viruses. For example, enzymes means integrase, reverse transcriptase, the function of these enzymes, capsid, you need to understand the function of the capsid, nucleic acid, genomes of the viruses, you need to understand their function. You need to understand the virus's structure and their function in a detail. So if you missed that video, so go back to my channel and watch the video about the viruses, their structure, their size, shape, everything the viruses. Once you get the idea, then the mechanism of the action of the antiviral drugs will be very easily for your understanding. So that's why before to understanding this antiviral drugs mechanism, first of all you need to understand the structure of the antiviral drugs. So go back to my channel and watch that video and then come back again and see this video about the antiviral drugs. Fine. So in this video we are just talk a little bit to summarize the antiviral drugs, that actually what we are going to talk. Fine. So now let me write. So now in this video we are going to talk about the antiviral drugs. Antiviral drugs. Usually we talk the overview or big picture, we can also say. Antiviral drugs, or we can say antiviral therapy, or we can say the antiviral agent. So there are different names. Fine. Now, first of all, what is antiviral drugs? Antiviral drugs are those drugs which are used against the viruses. So let me write. Used against the viruses. Because anti means against, viral means viruses. Fine. So antiviral drugs are those drugs which are used, which are developed, which are produced against the viruses. You know, these drugs doesn't mean just like, these drugs are not just like the antibiotics, because if you better know, the antibiotic means antibiotic are those agents which can kill or which can stop the activity of the bacteria. Fine. But whenever we are talking about the antiviral drugs, so antiviral drugs means that these drugs will not kill the viruses, just like bactericidal. It doesn't mean. It means that it can prevent the virus's replication. It can attack the virus's enzyme, attack the main component of the viruses. Due to these components, the viruses can replicate it. So once these components are inhibited by these antiviral drugs, so the viruses will be not replicated. So it means that we just stop the viruses from the replication. Fine. But on the other side, antibiotic means that there are some antibiotics which are used to stop the growth of the bacteria or to stop or we can say to stop the growth of bacteria and to kill the bacteria. Fine. So we'll be talking the antibiotic in the next video lecture. Fine. Is if we talking the basic overview of the antiviral drug, so you let me draw a host cell. Within the host cell, what are actually happening, and here I will just draw the virus structure a little bit, virus structure explanation, and after that then which kind of material that we should have to stop, which main component that we should have to stop inside the viruses. We'll be talking all in this video lecture, but in a short form. Now let me explain. Let's suppose this one area, let me draw also. Let's suppose this represents a host cell. Fine. Now let's suppose, consider that this one area represents the host cell, and this is a virus. Let me draw. Fine. Inside the viruses, the genetic material are also present. The genome of the viruses are also present. DNA, RNA, or any kind of the genetic material are also present. Outside the viruses, there are the spikes proteins, or we can say the glycoproteins are arises. GP, we also say that. We call that GP also. Fine. So now the viruses actually use these proteins. Once the virus use these GP proteins, so the viruses will easily attached and they will be enter their self, or due to these spikes or due to these glycoprotein, the viruses will make pores inside the host cell, by which the virus means it penetrate the host cell, or we can also say that it penetrate the cell by which the virus get enter. Fine. So how we can stop these things? Due to these things, the viruses will get enter, or we can say it can cause penetration. Let's suppose this hole means that it can cause penetration. That, so how we can prevent the viruses from this processes? So whenever we want to stop the activity of the entering and penetration, we give membrane fusion inhibitor. So what we give? First, we give the membrane fusion inhibitor. A membrane fusion inhibitor. In short form, we write MFI, membrane fusion inhibitor. And the drugs which are usually used in membrane fusion inhibitor, these are usually enfuviritide. Let me write the spelling. Enfuviritide. So we usually give these drugs whenever you want to stop the entry or the penetration of the viruses. You should have to give enfuviritide, because this enter and penetration means membrane fusion. So whenever you give the drugs against the membrane fusion, we call that membrane fusion inhibitor. And usually which kind of drugs we give? We give enfuviritide. These drugs, what actually do, we'll be talking later in the next video. Fine. Their mechanism of action, means the resistance activated against these drugs, will be talking in a later. Now, this virus also, let's suppose the viruses also show some resistance against these drugs. The virus also show resistance against these drugs. So how? We'll be talking in the next video lecture. Fine. So let's suppose the virus now show resistance against these drugs, and let's suppose the virus get enter inside the host cell. Then after that, what happen? You know, the virus contain a protein coat. The virus contain capsid area. Some viruses, when they enter inside the host cell, so they will just release the capsid area, and inside the capsid there are the genome present, the genetic material of the virus is present. So these things should be uncoating. Once the capsid area are removed, or we can say once uncoating process are done, then the replication process will be started. So we can say that after that, what happened? Uncoating. uncoating. You know, there are some also other drugs which can stop the viruses or which can block the viruses uncoating. So once we stop or once we block the uncoating phenomena, so the virus genome will be not released out. So once the virus genome will be not released out, so how the viruses will start their machinery? Sorry, not their machinery. How the viruses will use the machinery of the host cell. Fine. So the drugs which are used to stop this activity, usually we call that amantadine. Maybe the spelling is right or wrong, but maybe right, yeah. So we call that amantadine. Amantadine drugs usually available to stop the uncoating, mean the uncoating of the protein. Fine. And the protein means the uncoating proteins means the virus also show resistance against these drugs. How their mechanism of action and their resistance activity against these drugs will be talking later. After that, once the resistance is shown, then what the virus actually do? They will release their genetic material. Let me draw the genetic material with green. Let's suppose these are the genetic. They will release their genetic material, and once they release the genetic material, then they use the RNA polymerase enzyme. Remember? I hope you will remember this RNA if you read my lecture in the previous videos. RNA polymerase. So they use the RNA polymerase. Now with the help of this RNA polymerase, this genome will be converted into early proteins. Or from this genome, first we get the messenger RNA, early messenger RNA, and after early messenger RNA, we get the, let's suppose this red one represents early mRNA. So once the early mRNA are produced, then it will produce what? Early proteins. These black one represents early proteins. So how we can stop these things? How we can stop the early messenger RNA production and the early proteins? How we can stop this? We can stop these things whenever we are giving another drugs, and we call that fomivircine. Let me write So this fomivircine are actually used to stop the production of messenger RNA or to stop the production of early proteins. Fine, but the virus also show resistance against these fomivircine. How? Their mechanism of action and their resistance will be talking later in the next video. Fine. After that, once the virus show resistance against these drugs, then what happened? There are some other things inside the viruses. We call that nucleic acid synthesis. Nucleic acid synthesis. What does it mean? It means that nucleotides are formed, means that DNA and RNA formation. Nucleic acid synthesis means DNA or RNA formation. Fine. So these DNA RNA formation are also important for the viruses. Once these DNA RNA formation are done, then the capsid will around to the viruses and finally a mature viruses are formed. Not mature, but the capsid area of the viruses are formed. So we should have to inhibit this one area. So how we can inhibit this one area? We inhibit this nucleic acid synthesis by a drugs, and we call that reverse transcriptase inhibitor. So let me write. In short form we write RTI, reverse transcriptase inhibitor. Usually two reverse transcriptase inhibitor are actually happen. One we call that non-nucleotide reverse transcriptase inhibitor. Two drugs are used. Fine. One we call that non-nucleotide reverse transcriptase inhibitor, and the second we call that nucleotide reverse transcriptase inhibitor. Fine. So we usually give the drugs to stop the activities of nucleic acid synthesis, and these drugs are called as reverse transcriptase inhibitor, and these drugs are divided into two classes: non-nucleotide reverse transcriptase inhibitor and nucleotide reverse transcriptase inhibitor. Fine. Now in non-nucleotide reverse transcriptase inhibitor, the drugs are called atravirine, and for non-nucleotide, and the drugs which are used to stop the activity of non-nucleotide reverse transcriptase inhibitor, we call that lamivudine. We usually give these drugs to stop the activities of nucleotide reverse transcriptase inhibitor. Now, what does these two terms means? Reverse transcriptase enzyme are those enzymes which are used to convert the messenger RNA into a DNA. Fine. But non-nucleotide reverse transcriptase means that the addition of the nucleotide on the messenger RNA, by which we get a DNA RNA hybrid. So there are nucleotides which are, let's suppose this is a messenger RNA. So there are some nucleotides are added, and once the nucleotides are added on the messenger RNA, then what we get? We get a double-stranded, means sometimes we get a DNA RNA hybrid, or sometimes we get double-stranded DNA. Fine. So we can stop this activity by giving non-nucleotide reverse transcriptase inhibitor. Or other things, what does nucleotide reverse transcriptase means? It means the enzyme which are available to start the processes. Means which one enzyme? The reverse transcriptase enzyme. It means the reverse transcriptase enzyme. So the reverse transcriptase enzyme have a specific structure. We'll be talking later. They have a specific structure just like that, and the drugs will fit here and stop their activity. So once, let's suppose these are the drugs. The drugs are now present here, and they will stop the activity of this nucleotide, means of this reverse transcriptase inhibitor. Fine. So that's how. So that's all about the non-nucleotide reverse transcriptase inhibitor and nucleotide reverse transcriptase inhibitor. Their mechanism of action and the resistance against these drugs we'll be talking later. Now, once the resistance shows against these drugs, then what the viruses actually do? The virus produce late protein. The virus produce late protein. The late proteins, actually these are responsible for the structure. Fine. And this one protein, early protein, are responsible for the production of some essential enzymes for the further replication process, and this one late protein are important for the structure. These are actually structure protein, and these protein are responsible to make a capsid. So we should have to target this one late protein. We should have to target this. And which drugs are usually available to stop this activity, late protein activity? These drugs are usually called as we call that metazone. Let me write. Metazone. You know, when the proteins are made, so these proteins are very large, very complex, and the viruses cannot make their capsid with this large complex protein. So what the viruses do? The viruses produce a protease enzyme. These protease enzymes start to cut these proteins, and once these proteins are converted into a small protein section, then the viruses will make a capsid from this small protein, from these structured proteins. So we should have to inhibit these protease activity, and this protease activity will be inhibited by the methoxazole. Methoxazole, their mechanism of action and their resistance will be talking in the next video. Fine. After that, the virus also shows some resistance against these methoxazole. And once the virus shows the resistance against this methoxazole, then what happened? Then assembly of viruses. Assemblies of viruses are occurred, means the viruses are no formatted, or we can say the viruses are no formed. So we can also stop this formation, the viruses formation. And how we can stop it? We can stop it with a rifampin. We can stop the assembly of viruses with a rifampin. So we give the rifampin drugs by which the assembly of viruses are stopped. Fine. But viruses also show resistance against these drugs. The resistance and the mode of action of these drugs will be talking later. Now after this, once the resistance shows, then a mature viruses are formed. Let's suppose a mature viruses. Let's suppose this is a virus is having the genome and every materials are present. Then what actually, let's suppose this is a virus, fine. Let's suppose the viruses are made. Whenever they are released out from the host, so they will release some enzyme from their surfaces, from their surface area, from their glycoprotein area. They will release some enzyme, and we call that neuraminidase enzyme. Neuraminidase. The neuraminidase enzyme, what actually it does? This neuraminidase enzyme will burst the cell, or sometimes this neuraminidase enzyme are also used for the cell bud for the to release out the viruses through a cell bedding, or this neuraminidase enzyme are also used to lyse the cell and to release the viruses. These neuraminidase are used for this purpose. So once we stop the activity of the neuraminidase, so the viruses will be not released. Although the viruses are formed, but they will be not released from the host cell, and there the viruses will not do their life cycle, and they are not able to kill the other or to start their life cycle again on the other host cell. Fine. So how we can stop this activity of the neuraminidase? We can stop these neuraminidase when we give the drugs, and these drugs are usually called zanamivir and oseltamivir. So now let me write the name: zanamivir and oseltamivir. So these drugs, once we give, so they will stop the activity of the neuraminidase enzyme. So the neuraminidase activity should be stopped by these drugs, should be blocked by these drugs. So this is a basic overview of the antiviral drugs. Once you get the brief idea about the antiviral drugs, once you know that which kind of drugs that we are going to talk in the next video, it will be very easily for your understanding. But as I told you, whenever I am starting this topic from the beginning, as I told you, that you need to understand the virus's structure, their important component, the function of these important component. Fine. Once you know the idea, so the mechanism of action and the resistance will be very easily for you to understand. So go back to my channel and watch the structure and watch the important component, mean the messenger RNA, their replication, the life cycle of the viruses. You need to know all these things. Then the rest of the things will be very easily. Then come back to this video and watch the video of the antiviral drugs, their mechanism of action in a detail, by which you will very easily understand. Fine. So in the next video we will talk the membrane fusion inhibitor. different drugs which are included in the membrane fusion inhibitor, the resistance which are activated against this membrane fusion inhibitor, and also the mode of action of these drugs. Fine. So stay tuned and continue to watch. Thank you.